Assalamualaikum and a very good morning to everyone. This is Group 8's presentation on a BMC Genomics paper about unraveling the genetic traits and functional diversity of the pan-genome in Pantoea dispersa. My name is Christina and these are the other members of Group 8. Cotton swabs are essential tools in a medical setting, usually used for cleaning wounds, applying medication, and also collecting samples. However, if it's not properly sterilized during manufacturing, it can carry harmful microorganisms, which serves as a vector for pathogens, posing significant threat to a patient's health. One concerning example is Pantoea, a genus of bacteria commonly found in water, soil, and plants. While some species can cause plant diseases, Pantoea can also infect humans, where in severe cases, it can cause sepsis. Despite advancement in medical research, how Pantoea can cause disease in humans is still not fully understood, and research investigating a known species of Pantoea, which is Pantoea dispersa, still remains limited. Due to its diverse biosynthesis and biodegradation abilities, Pantoea exhibits a significant value in agriculture, clinical application, and environmental remediation. For example, Pantoea dispersa, which is widely present in rice seeds, can promote plant growth by producing IAA and other plant hormones. As for Pantoea aldomerans, it has been demonstrated to inhibit the growth of fungal pathogens and also isolated low molecular weight lipopolysaccharide IPPA1, which exhibited broad therapeutic properties, holding great potential in prevention and treatment of diseases in humans and animals. This study sequenced 3 strains of Pantoea dispersa isolated from a batch of microbiologically contaminated cotton swabs for analyzation in conjunction with existing data from 57 strains of Pantoea dispersa found in NCBI. The objective of this study is to analyze the phylogenetic relationship and population structure characteristic of Pantoea dispersa strains, to construct its pan-genome framework, to evaluate the industrial and clinical implication, especially of its secondary metabolites, and also reference for the clinical treatment of infections from Pantoea dispersa. The last objective is to investigate the distribution, characteristic, and functional genomic traits such as the virulence factor and antibiotic-resistant genes, and also the secondary metabolites genes clusters.
Next, I will be presenting about the methodology. So the first one is isolation and cultivation of Pantoea dispersa. Pantoea dispersa was isolated from 3 swabs that failed sterility test. Swabs were incubated in thioglycolate and soybean casein broth at 33 degrees Celsius for 3 days. Colonies were grown on cryptic soil agar and purified. The next one is DNA extraction and genome sequencing. DNA extracted from cultured cells using metagenomic kit. Library quality checked via qubit and QRT-PCR. Sequencing performed using Illumina MiSeq platform to generate 150 base pair, paired, and reads. And the third one is genome assembly and annotation. Reads trimmed with Trimomatic, then assembled with spades using multiple K-mers. Quality evaluated using QUAS, and species confirmed using BLAST and GTDP-TK. So the next step is whole genome phylogenetic analysis. 57 reference genome used from NCBI. NSPs filtered and concatenated using BCF tools. 3 constructed using RAXML and visualized with iTool. ANI calculated using FastANI. Next is population structure and genetic diversity. Structure used to determine clusters K using BIC. Diversity indices such as nucleotide diversity, fixation index, and Tajimas D calculated via VCF tools. Nucleotide diversity measures polymorphisms. Fixation index measures population differentiation, and Tajimas D indicates evolutionary pressure. So for the second last method is pan-genome analysis. Orthologous groups, OGs, identified with orthofinder, genes classified as core, soft core, shared, or cloud. Longest protein per OG selected as representative for function analysis. So for the last one is functional annotation. VFDB used to find virulence factors. CART database used for antibiotic resistance genes. And the last one is anti-smash and BigScape used to identify and cluster secondary metabolite biosynthesis genes. So that's all for the methodology. Thank you.
In this study, we analyzed 60 Pantoea dispersa genomes, including 3 newly isolated strains, to explore genetic diversity, population structure, virulence, antibiotic resistance, and secondary metabolite potential using a pan-genomic approach. We begin with the de novo assembly of the 3 new isolated P. dispersa strain and 57 existing strains from NCBI. In Figure 1A, shown that the visual comparison of all 60 strains to the reference genome LSCH. Major gene deletion were observed at position 600 kb, 700 kb, 1300 kb, 1500 kb, 2900 kb, and 3400 kb. Based on Figure 1B, the graph shows the cross-validation error for the different number of population cluster. K equal to 4 has the lowest error, indicating that the strain fall into 4 distinct genetic clusters, which are P1, P2, P3, and P4. In Figure 1C, the 60 strains were divided into P1 that has 7 strains, P2 7 strains, P3 32 strains, and P4 40 strains, validating the population structure. As seen in Figure 1D, the degree of sequence variation within each cluster were displayed. P3 has the higher diversity, while P2 has the lowest, suggesting clonal similarity. In Figure 1E, FST measure genetic divergence between cluster P1 and P2 are the most genetically distinct, while P3 and P4 are the least, showing they are closely related. As seen in box plot of Tajimas D in Figure 1F, P1 and P2 show neutral evaluation. while P4 indicates population concentration or loss of rare allele. In figure 2a, the phylogenetic tree was built from 28,704 SNP. The tree showed the evolutionary relationship among the 60 strains. For figure 2b, P2 cluster strains show nearly 100% ANI, suggesting a clonal group. We construct the pan-genome, which we call PDSPAN, and identified 6,791 autologous gene cluster. As shown in figure 3a, only 45% were core genes and the rest were accessory, suggesting high genomic flexibility. In figure 3c, core protein are longer than accessory one. That suggests core protein are more conserved and likely functionally essential. Based on figure 3d, shows that blue curve, which are pan-genome, increase with more strain. That means the pan-genome is still expanding, and the red curve, which represent core genome, gradually decreasing with more genome added. Next, we look at the virulent factor. Figure 4a showed how virulent factor and antibiotic-resistant genes are distributed across the 4 clusters, and it showed that the cluster P3 and P4 are denser in virulent factors. In figure 4c, P3 has the most virulent factor, especially those who related to motility, nutrition or metabolism and immune modulation. In figure 4e, we also identified 12 antibiotic-resistant genes with 9 conserved across strains. However, SUL1 gene, sulfonamide resistant, appear in only 2 strains.
Next is figure 5 for the biosynthetic gene cluster or BGCs, which are responsible for the production of secondary metabolites. Across 60p dispersa strains, we identified a total of 289 BGCs. Out of these, only 23 clusters match known types such as the 4 carotenoids and siderophores. This vast majority, 266 clusters, were novel discovery. About 30% of these novel BGCs were predicted to be aryl polines, which are known for their antimicrobial properties. Other major types included non-ribosomal peptide synthetase, NRPS, terpenes, and RIPP-like cluster, each making up around 20%. The smaller proportion included RRE, nucleoside, thiopeptide, and SI amino acid types. Next, group of BGCs into 18 gene cluster family or GCF. 5 of this family, NRPs 1 to 6, terpene 1 to 5, RIPPS 1 to 7, others 252, and others 253 accounted for over 80% of these overall clusters. Among these, the RIPPS 1 to 7 family showed the densest network connection, which suggests a high degree of evolutionary conservation and functional importance within the species. Then, they analyzed the conservation of BGCs related genes across the pan-genome. So these genes were classified into 227 orthologous gene clusters. Of these, 158 were core genes found in all strains, 20 were soft core, and 43 were shell, and only 6 were cloud genes. Thus, this means that most BGCs are highly conserved, indicating that secondary metabolite production is a fundamental and stable feature of P. dispersa. Next is figure 5 for the biosynthetic gene cluster or BGCs, which are responsible for the production of secondary metabolites. Across 60p dispersa strains, we identified a total of 289 BGCs. Out of these, only 23 clusters match known types such as the 4 carotenoids and siderophores. This vast majority. Moving on to the discussion, first we highlight the ecological duality of P. dispersa. So this bacterium is found in diverse environments, including plants, soil, animals, and even clinical settings. It plays a dual role, which on one hand, it benefits plants by solubilizing phosphate and producing plant hormones, while on the other, it can act as a pathogen, causing infections such as sepsis for the newborns. So next, the analysis of the genomic diversity and evolution show that P. dispersa has a large accessory genome, which is almost 54.9%, and a substantial core genome, about 45.1%. So this combination indicates both genetic stability and adaptability. So the high level of single nucleotide polymorphism and open nature of its pan-genome suggests that P. dispersa is continually evolving and adapting to various environments. Looking at secondary metabolite potential, they identified 289 biosynthetic gene clusters, the majority of which are novel. This includes arylpolines, which have antimicrobial properties, and NRPS metallophosphates, important for ion acquisition and microbial competition. So the high proportion of core BGCs genes suggests that producing secondary metabolites is a fundamental and conserved features in this species. So it is also found that a diverse set of virulence factor, which is 406 in total, distributed unevenly among strains. So most are linked to metabolism, motility, and immune modulation. Importantly, the presence of type 3 secretion system gene supports the potential for both plant and human pathogenicity, with some virulent factors unique to specific gene clusters. In terms of antibiotic resistance, they discovered 12 resistance genes with 9 highly conserved across all strains. This mainly confer resistance through antibiotic efflux and target alteration. The widespread presence of these genes suggests that P. dispersa may have robust antibiotic resistance, which could complicate clinical treatments and needs further investigation.
In conclusion, this study explores the genome of Pantoea dispersa, which reveals its genetic diversity, antibiotic-resistant genes, virulence factors, and also its potential to produce antimicrobial compound, which help us to understand better how the bacterium adapts and can cause infection, especially in clinical settings. I also like how it raised questions to myself due to its dual behavior, where it has benefits to plants but can cause disease in humans. By identifying the key resistance and pathogenicity genes, this research could support better infection control and also contributing to developing new antimicrobial agents, where it will help to ensure patient safety and public health.
So for For my part, I think the presentation provides a clear and concise overview of the study's main findings, particularly the construction of the first pan-genome of Pantoea dispersa. It effectively highlights the genetic diversity observed across the 60 strains and the significance of accessory genes in shaping functional variability. The inclusion of virulence factors and antibiotic resistance genes adds depth and relevance, especially in a clinical context. Mentioning the open nature of the pan-genome would further underscore the potential for ongoing discovery in this area. Overall, the presentation communicates the study's contributions and implications with clarity and focus. So that's all from me.
As for me, I think the study presents the detail of pan-genome analysis of Pantoea dispersa, which help to understand how genomic data can be used to explore bacteria diversity and function. The discovery of a large number of accessory genes, virulent factors, and antibiotic-resistant genes show that P. dispersa is not only adaptable but also potentially pathogenic. There are also secondary metabolic gene clusters, which suggest possible application in biotechnology. Overall, I think the study shows how genomic can be applied in both research and real-world contexts, such as healthcare and industry.
So for me, Pantoea dispersa is a genetically diverse and ecologically versatile species with significant potential for both beneficial applications and also pathogenicity. It is because its rich collection of virulence, resistant and secondary metabolite genes that makes it an important organism for the future research in medicine, agriculture and also biotechnology. So I think that's all from me. Thank you.